I do believe it's feasible. We're not the first in this field or in this niche, trying to establish gene therapy programs for AD. As I said, I just came back from St. Louis and from AAC conference, and there was fairly small conference which focused only on APOE, including APOE treatment, APOE therapeutic program, and it was very interesting to me to see the development of this field. So there's many other approaches goes in parallel to what we're trying to achieve, and it's very encouraging. For example, there was a report from a PI from MIT, and she's also a co-founder of Atalanta, a therapeutic company in Boston, and her name is Anastasia Horova, and she used a siRNA or RNAi program or RNAi tools to downregulate APOE, and she demonstrated very impressive results with APOE downregulation. There are other programs or other investigators at this conference reported different approaches using active Cas9 or RNAi, as I mentioned to you, antibody-based approaches. So it's not that field is completely empty. There's very encouraging developments. We may be biased, but I truly believe that our approach has significant advantage over other approaches. And indeed, for example, comparing to siRNA, what was used in the approach I mentioned to you, the reduction of APOE, it was super impressive, but it was almost 100%, and you not necessarily want that level of downregulation or knockout in this case, because I mean, in some cells, APOE express at very low level, for example, in neurons. So this approach will completely kill APOE in those cells, and we all know that we still need to have some physiological levels of APOE expression. And so this is one of the disadvantages of RNAi approach. You don't have good control over the capacity. It has been demonstrated also in other studies not related to Alzheimer. For example, for Parkinson's disease, this approach found to be toxic because it just too robustly down-regulate expression of protein of interest, and it's not necessarily a good, not necessarily an advantage. So our approach allows for what we call fine-tune regulation. The fine-tune regulation, we know exactly that there's flexibility of this approach to bring a repression of gene of interest to a certain point, and we know how to achieve this. Same with other approaches. And again, there's no ideal approach, but we do believe that combined effort and recognition that APOE is a very important target as most significant and reproducible risk for a late onset of Alzheimer's disease. Recognition and not necessarily extremely encouraging results with other targets such as tau or amyloid.